Assalamu alaikum. I hope so you're all doing fine. I'm Dr. Muhammad Ali Rabbani, and today we are going to discuss the next lecture in the series of journal embryology, that is oogenesis and female reproductive cycle. I know this is a tall order, and there are a lot of things to be discussed here. The oogenesis and follicular development, and then endometrial changes, and the hormonal levels, and the interrelationship between them. So there are a lot of things to be discussed here. I will try to make it as simple for understanding as possible, and I will try to uh, make the chapters down in the that you can jump to, so that you can jump to a particular part. So with that, let's start with our learning objectives. By the end of this lecture, the student should be able to describe the pre and post natal maturation of oocyte, describe the process of ovulation and Schmidt's pain, explain the significance of meiotic arrest, describe the formation, function, and fate of corpus luteum, define menstrual cycles and its phases, and describe the steps and hormonal control of female reproductive cycle. That if I have not mentioned previously, I'm borrowing these learning objectives from the UHS curriculum, so that I'm sure I'm confident that each of these things are covered. If you are in a university that has curriculum and have learning objectives that are not covered in the lectures, do mention in the comments below. I will try to adjust them as I go along. So let's start with what is oogenesis. Oogenesis is the process by which oogonia develops into the mature oocyte. Oogonia is basically the female primordium germ cells. So this occurs in ovary, begins before birth, and is completed after fertilization. That's the different part. So let's not dwell on the different thing. I will have a separate video on the differences between spermatogenesis and oogenesis. So this is the thing that I think, discussing with you for the third time now. We discussed this in the before the cell cycle. We discussed this in spermatogenesis, and now in this oogenesis. Primordial germ cells are the cells that are going to form the oogonia. So these cells are initially in the epiblast. They migrate through primitives in the week 2. Second week, they migrate through primitive streak to yolk sac in week 3. And in yolk sac, they are present in the tail end where the allantois is there, the outpouching of the yolk sac. From here, the yolk sac is going to form the gut. The hind foregut is here, the hindgut is there, and it goes through the gut wall. It goes through the mesentery. Mesentery is the part that is joining the gut with the posterior abdominal wall. It crosses the gut at week 4. It reaches the genital, reaches the future gonads about in week 5, and it enters or invades the genetical ridges in week 6. And basically, after the invasion in the week 6, the signal induction helps the differentiation of the indifferentiated gonad into males, testes in females, ovaries. So this is the point where this different occurs. So oogonia. Primordium germ cells, they divide all the way, more than one divisions, many divisions, all the way till the primordium germ cells divide to form oogonium, and they divide again by a number of mitotic divisions until they form the primary oocyte. So this is the part, all of this before it occurs through mitosis. Primary cell, you have to keep in mind this cell, primary oocyte, all the way till primary oocyte, the division occurs through mitosis, however number of divisions are there, and after that you will enter into meiosis 1. So let's put a pin in that. Let's focus on the part we will behind. So primordial cells divide and differentiate into oogonia, and those divide and give rise to primary oocyte. And during the division process, not only they divide to form more than one oogonium and more than one divisions for prime. This step replenishes. One stem cell goes back to the prime stem cell pool, and the rest goes to oogonium. So they increase in size. They reach about 600,000 by second month, 7 million by fifth month. By the way, these figures are a little controversial. I am quoting the figures from the Langman's Embryology. So if you read different books, you will have a little different figures. But in the ballpark you will have again in the hundred thousands and the millions for the second and fifth month respectively. Okay, now interesting thing that happens is that in males you had the stem cells that were surviving and continuously producing that. In females, by the fifth to seventh month, this primordium germ cells, they are spent. There is no reserve of the stem cells, and no reserve of the stem cell means that ultimately all will be in the primary oocyte stage, so they are not replenished. That is why female gametes and female eggs are very precious, because they are produced once before birth, and after the maximum point is reached by 7 million by fifth month, their number is then reduced from here onwards. So we talked about primary oocyte that's formed by this formed by mitosis, and this will undergo meiosis one. So in the embryonic life, in the intrauterine life, all of these mitosis occurred, and they form the primary oocyte, and this primary oocyte started in meiosis one. But that interesting thing happens. In the meiosis 1, it has to go through prophase 1, metaphase 1, anaphase 1, and telophase 1. And in the anaph prophase, there was leptotene, zygotine, pacatine, diplotin, and then there is dikinesis phase. For the details of these different phases, do refer to the lecture on meiosis that is present in our lecture series in the playlist. So, in prophase 1, in the diplotin phase, the meiosis stops. The cell is arrested at this point, and there is no progressing to the next phases for now. So the ovum starts to fill up with the primary oocytes that are stuck in the diplotin stage of the prophase 1 of meiosis 1. And this is maintained by an um, factor that is called oocyte maturation inhibitor OMI, and this stage is maintained all the way till puberty. And all of these cells that are now arrested in this particular phase are called primary oocytes. And that's why this cell stuck in the prophase 1 diplotin of prophase 1 of meiosis 1, this will be carried all the way till puberty, and not all the way till puberty. From the last ovulation that occurs before menopause, this is was arrested at the intrauterine life. So the fact that risk of having children with chromosomal abnormalities increases with maternal age, because that oocyte, primary oocyte, was put in reserve for more than 50 years from the intrauterine life, so it has more chance of getting corrupted or having an influence from the environment. So we have primary oocyte. Oocyte was the ovum or gamete. This is frequently confused with the follicle, and please do not do that. So each oocyte, this was the oocyte, this was the oogonia. Oogonia were further dividing until they reach the prophase of meiosis 1. Okay, this is the ovum. The yellow shows the interstitium. This orange or brown, this shows the epithelium of the ovary. So some epithelial cells of the ovary, they break off and they start to encircle this pool of multiplying oogonia. So these are flat epithelial cells, and with time these cells divide in such a way that one cell encloses this, one cell encloses this, one cell encloses this. So you will have one oocyte, primary oocyte, stuck in prophase 1 in meiosis 1 diplotin stage, and surrounded by one or more follicular cell. So this combination of primary oocyte and the flat epithelial cell or flat follicular cells, so flat supporting cells shown gray here, so this combination of these two things is equal to, this is called the ovarian follicle. So whenever ovarian follicle comes in mind, you should think of two things: the oocyte in the center, the primary oocyte, and surrounded by the flat follicular cells, those are the ovarian follicular. Now these flat cells are called follicular cells. So primary oocyte surrounded by flat follicular cell is equal to ovarian follicle. These two things. The primordium and this oocyte, this was primary germ cells in origin. Ultimately, it was coming from epiblast, and this the follicular cells are the originating from the epithelial covering of the ovary. So there is one primary oocyte per follicle, and they represent the ovarian reserves. By as you discussed in the previous slide, by the seventh month, fifth month, you will have 7 million, but now they start to die. They undergo atresia, and their number steadily decreases. So by the seventh month, most of the oogonia have been transformed into the primary oocyte. By the end, you do not have any primordium germ cells reserve. You don't have any primordial germ cells reserve, you don't have any oogonia, all of these things have gone, and now you all the what remains is the primary oocyte. These oogonia develop into primary oocyte before birth by seventh month, and they're continuously lost by the atresia, and this is basically the nature's mechanism of getting the best out of it. So they reduce it so much so that 90% are lost roughly, and out of those 7 million you get at birth, 6 to 800,000, you get an average of 700,000 at birth. There is a difference little in Langman that says 6 to 700,000 and KLM says 2 million, but still when you are talking about 2 million, you had a drastic drop from the 7 to 8 or 10 million that was in the beginning. And this atresia process does not stop in pre-birth, and it continues after the birth till the puberty and the time till the female starts her reproductive cycle. So at the puberty, it is about again 90-95% are roughly lost. You start from 800,000, you get to 40,000, you have 90% decrease. So 95% decrease. So only 5% survive. So this is the 40,000 oocytes. And from the 40,000 oocytes, if you figure it out, let's say a woman was fertile from age of 15 to age of 55. So there is roughly 40 years between them. And if she ovulated all in each and every of those months of her cycle, you get about 480 oocytes. So all of those you get 480 oocytes that are released by ovulation. So they are further reduced during pregnancy or oral contraceptive failure, anything event that occurs there. So that is a nature mechanism of getting the best out of the situation. So up till now, we have discussed what were primordial germ cells, where they originated, how they reached there, what happened with them, and you now have an ovary at the puberty. Ovary is filled with the at puberty follicles, primary follicles. Sorry, primordial follicles. I will discuss the detail of follicular development shortly. And primordial follicle consists of the primary oocyte stuck in diplotin stage of prophase of meiosis 1 and surrounding follicular cells, and it is stuck there. There is no multiplication or division and growth is going on, and everything is stuck there. Roughly you have 40,000, and now what will happen at the puberty? So what happens in the puberty? You have this is a picture. You can see this events from different vantage point. You can see this from the events of the follicle development. The primordial will convert to primary, secondary, vesicular. So the follicular growth phase is called follicular phase. And you have ovulation, and after ovulation, we have left behind this corpus luteum that initially grows and then regresses. So you have luteal phase. Same events can be seen from the aspect of gonadotrophins. You have follicle stimulated hormone, luteinizing hormone. In the first phase, you have follicle stimulating hormone that dominating. So that can be, and then you have LH surge, and then in the next phase, you have luteinizing hormone dominating. The same events can be seen from the level of reproductive hormones, the estrogen and the progesterone. The first phase, the initial half up till this point, is also called estrogen phase because that is the one dominating. And after this, after the ovulation, the luteal phase is also called progesterone phase because that is the dominating one. And you can see all the same thing again from the perspective of the menstruation or from the perspective of endometrial changes. Initially, within the follicular phase, you have menses time, menstrual phase, you have the growth and repair time that is called the proliferative phase, and during the follicular phase, you have the secretory phase where the endometrium secretes a lot amount of fluids and is ready for implantation. So you have these different states from the different lenses. So here we will be focusing on follicular development and gonadotrophin developments a little more. So but just you can see that follicular phase from the start of the cycle. So day one is the day one when your bleeding starts, when the menses start. The follicle will the primordial follicle. You start with a primordial follicle. It goes through the stages of primary, secondary, vesicular, until Graafian follicle, and until it goes to ovulation. So all of the different stages of follicular development. So this first half all the way till ovulation, it's called the follicular phase. We will see in detail each of these steps. And here the follicle stimulating hormone is the dominant one. Ovulation is the point where ovum is expulsed out of this follicle, and this is triggered by high level of luteinizing hormone that is called LH surge. And after that you have in the ovary remaining the follicle without the ovum. So this is called corpus luteum, and it grows initially and then regresses all the way till corpus albicans, and those different phases are summarized under luteal phase. So from the perspective of follicles, the cycle reproductive cycle can be divided into follicular phase. It can be divided into follicular phase, that is of the growth and after the ovulation point, and then there is luteal phase, and then there is follicular stimulating hormone in the first one and luteinizing hormone. Let's see how this works. Again, these lectures are discussed in much more detail in the reproductive physiology lectures. Technically you will be discussing them in the second year as per our syllabus, but if you are studying them now, you can refer to those lectures too. Follicular development ovarian follicle consists of oocyte. I'm reiterating it again and again because I have seen students making this mistake. They cannot differentiate between oocytes and follicles. So do make a point of that. Ovarian follicle is equal to oocyte surrounded by one or more layer of epithelial layers. So epithelial layers, these are the basically flat cells, follicular cells. So follicular cells plus oocyte is equal to this ovarian follicle. So when the female gets to the puberty, these FSN let's start to circulate in the body. They were not there previously, but now they are there, and then they go through different stages. The primordial follicles, then there is growing follicle, primary and secondary, and then there is the mature follicle, the antral and vesicle, and then there will be mature, just pre-ovulative follicle that is called mature and Graafian follicle. So what is primordial follicle? The one that we studied in the previous step at the reserve stage, that was the primordial follicle. The primordial follicle consists of primary oocyte stuck in prophase 1 of meiosis 1, and in the prophase 1 there is diplotin stage, and it is surrounded by a single layer of flat epithelial or flat follicular cells. So this was the primordial follicle. So it started from the third month, and by seventh month all were in the this phase. All follicles were in primary follicle stage, and all eggs were in the primary oocyte stage. So you had all of these at the seventh month. So this is the primary oocyte, flat cell surrounded by primary oocyte, diplotin stage of meiosis 1 surrounded by flat single layer follicular cells. Just reiterate which was oocyte stage, diplotin stage, prophase 1, meiosis 1. Now, the primary follicle. Now, the FSH is the FSH follicular stimulating hormone. As the name indicates, that it stimulates. As you can see, follicle is level is high. The follicle stimulates the growth of primordial follicles. It does not stimulate all of the follicles. As the puberty, after puberty, it reaches 15 to 20, some books says 20 to 30 follicles, primordial follicles are randomly picked, and they start to grow, through different phases, and ultimately one dominates and reach the full maturation, other go under atresia, and that is called corpus atreticum. So let's see what happens in those that do mature. Now, how is primary different from primordial? Two things. One, this follicular cell, they shifted from flat to cuboidal. As you can see here, you had flat follicular cell in the primordial. Now they started to become cuboidal in shape. Initially, so when they become cuboidal in shape, you rename them to primary follicle rather than primordial follicle. And moreover, the oocyte from the inside and the follicular cell from the outside, they secrete this material, the acellular material that gathers in between them, that is called zona pellucida, majorly consisting of glycoprotein. As the growth continues, these follicular cells that are forming the single layer, they make up multiple layers. So from single layer they go to stratified. So even it goes from single to the stratified layer, these are called primary follicle. So you have single layer and then stratified primary follicle. Okay, and now one thing happens, these cells after the stratification especially, the individual cells are no longer called follicular cells, these are now called granulosa cells. So keep in mind this change of name that is continuously occurring. For initially there were the surface epithelial cells of testes, then they become the flat epithelial cells covering the follicle, then they were called follicular cells. Now as they increase in size, increase in number, and they form a large population, they are now, the same cells are dubbed now as granulosa cells. So granulosa cells, location of the basement membrane is on the outside. Here is the basement membrane on the outside, and it abuts with the basement membrane of the cells that are gathering on the outside here. So basically these are the theca cells. The oocyte was coming from primordium germ cells. These granulosa cells were coming from the surface epithelium of the testes, and then there are cells that start to organize on the outside of the granulosa cells. These are called theca cells, further divided into theca interna and theca externa. These are basically the ovarian stroma that surrounds it. So theca interna is basically secretory function. It provides steroid hormones and the provides androgens for the circulation. And theca externa is basically fibrous one, fibrous support that gives support to the whole of the ovary. Theca cells are thought to produce angiogenesis factors that promote growth of the blood vessels in the theca interna, and that further supports the nutrition of the follicle. So this is about the primary follicle. Do you remember what stage oocyte was on? Yes, the same one. Diplotin stage of prophase of meiosis 1. Now the secondary follicle. Now the secondary follicle is identified this creation of the spaces within the granulosa cell. As you can see, the starting of the spaces start to appear. So this is now changes its name to the secondary follicle. When the flat converted to cuboidal, the name changed to primary. Remained primary as it become multilayered. But when fluids start to accumulate and the follicular fluid or liquor folliculi make spaces appear between the granulosa cell. Now this is called secondary follicle. And then these spaces, as they grow, they begin to coalesce with one another to form larger and larger space. And this large common space is now called antrum or vesicle. Some books they say that the secondary follicle is defined when antrum is formed, and I'm following Langman here, and that says when those spaces are formed, that is called follicles. Then you have granulosa cell. They are there. They are their function is to support the developing oocyte, and their function is to produce estrogens. Uh, yes, estrogens, and they're producing. And the theca cells. I told you this was theca, and these were secretory cells. They used to produce precursor dihydroandrosterone, and this is converted into estradiol in the granulosa cells. So basically secretory function, the hormonal function is doing conversion of androgens and the production of estrogens. The theca cell, this one, the theca interna, this is a secretory function, it produces androstenedione, basically not dihydroandrostenedione, androstenedione, and it produces testosterone too. Yes, testosterone is produced in female. So these androgens are produced by theca interna cells. This androstenedione is shifted to granulosa cell that converts into estradiol and so on. And theca externa is in the supportive function. Oocyte stage, diplotin stage of prophase of meiosis 1. Then enteral or vesicular follicles. Some books like Langman's consider them as the mature, rather developed stage of enteral follicles. Some books uh, separate it as after secondary follicle, and some consider it a part of secondary follicular stage. So don't worry, the basic crux is the same. As the same collects to form the same common cavity that is called antrum, so now this is called the antral follicle. It enlarges. It starts to enlarge and until it reaches the mature size, and that mature size is about 25 millimeter. 25 millimeters mean 2.5 centimeters. So yeah, 10 millimeter is 1 centimeter, so 2.5 centimeters. So this is quite big, about 1 inch. So it is very visible with the naked eye, and you have a large vesicular space. You have granulosa cells surrounding all around. You can see the ovum now protruding into the antral cavity, like here through the granulosa space, and you have well-defined theca interna and theca externa. Theca interna with its secretory function, and theca externa with its supportive function. Now, I told you the follicular surface epithelial cells change their name to follicular cells, they change their name to granulosa cell, and now I will introduce two more names of the same granulosa cells depending upon their location. So there is cumulus oophorus and corona radiata. As you can see, the granulosa cells around the ovum, they form a growth, or they form a mound that projects into the antrum. So these cells as a whole, these are called cumulus oophorus. And of those cells, the cells that are more immediately or more intimately near the ovum, surrounding the zona pellucida, the innermost granulosa cells, those that follow the ovum after ovulation too, those are called corona radiata. I think there is no hard and fast demarcating point at this stage. So some books demarcate all of them as corona radiata, some books demarcate all of them as cumulosuphorus, or you can differentiate that or the whole mount as cumulosuphorus and the most intimate one as corona radiata. So now we are ready for ovulation. So up till now we have not discussed LH. So up till now, before it, we are having in which stage? Again, prophase 1 of meiosis 1. This is the largest of the stage, compassing about 37 hours prior to ovulation. So as you can see, this is the laparoscopic image of the ovary. As the ovum, as the follicle becomes such large, 25 millimeters or 2.5 centimeters, it actually pokes a protrusion on the surface of the ovary. This protrusion on the surface of the ovary, this is called stigma. This is the bulge created by Graafian follicle, and that is how you can identify also. This is the point where you will have the ovulation, and this information comes in handy where you want to aspirate ovum for assisted reproductive techniques. So now what is ovulation? Ovulation is basically the hormone mediated process by which oocyte is released from the ovary. It usually occurs 14th day of typical 28-day cycle. Usually it is 14 day before the menstruation. So this figure 14 day before is pretty constant figure, and depending upon the variation in cycle size, for example, a woman has a smaller cycle size, let's say 24 days, then this period will be short relatively. Rather than we having a 14 days, you will have 10, 12 days. If the woman has a very last cycle, that is 40 days, so you get to 24 days. So this period before ovulation is variable, and the ovulation till menstruation, this is a pretty constant, stable period. In humans, only one oocyte is liberated during each cycle. Yes, there are instances where more than one are to and then lead to twins, and especially more than one release is a situation possible when there is hormonal trigger is given. So just before ovulation, you have this whole surge of increased level of luteinizing hormones. This occurs about a day preceding. This occurs. This usually occurs about a day preceding the ovulation. So this occurs here before day preceding the ovulation. Here is the ovulation. So it occurs here, and it does a lot of things. So what does luteinizing hormone surge does? So luteinizing hormones triggers a sequence of major events. Up till now, I have been iterating again and again that oocyte, regardless of the stage of follicle development, regardless of whether it was primordial or primary, single-layered, multi-layered, or secondary, or enteral, or Graafian, it was the oocyte was stuck in the prophase 1 of meiosis 1. But this LH surge, this increase in level, this triggers the meiosis 1 completion. It moves along from the prophase 1, goes to metaphase, anaphase, telophase, and does completes the meiosis 1. Then you have secondary oocyte with reduced chromosome number and one polar body formed, and polar body is given in the perivate line space, the space between ovum and the zona pellucida, and it does enter into the second meiosis 2. It goes through prophase and goes into metaphase, and then it gets stuck for the second time in the metaphase 2, and this occurs just before ovulation, about 3 to 4 hours before ovulation. So now you have the ovum before ovulation is ready to come out, now stuck in metaphase 2 of meiosis 2. Now the other things play their part. The granulosa cells are stimulated to produce hyaluronic acid, prostaglandins. Hyaluronic acid loosens the granulosa cells and it imbibes water. It increases the fluid volume in the antrum cavity, so pressure increases. Then prostaglandins are released and they trigger smooth muscle contraction around the in the thick externi especially, and they again increase the pressure further. So you have a very full follicle, pressure is increasing constantly. So if you increase pressure beyond that point, until there is going to be rupture, and the ovarian wall is weakened too. Plasminogen factor, activated plasminogen is released from the broken capillaries as the pressure is pushed against the ovarian wall. The broken capillaries release plasminogen. The surface of the ovary, that point is weakened. The stigma point is weakened, and this active plasminogen degrades collagen, tunica, albuginia, and surface epithelium are affected. So you have a weak ovarian wall, high pressure in the follicle, and all of this generated pressure, high pressure, weakened wall, surface ruptures at the point of stigma. So the oocyte with zona pellucida surrounding, with immediate surrounding granulosa cells that were called corona radiata, these were granulosa cells. So they are expelled. They are expelled. So oocyte immediately after coming out with the follicular fluid, with the zona pellucida and those, they initially adheres loosely to the ovarian surface because the follicular fluid helps it and the thick viscous fluid helps it. Then the ovarian follicles, the ovarian tube, it's like the fimbria, they are sweeping across the surface of the ovum, and it uh, picks up that ovum, that follicle, the ovum, not follicle, the ovum, it enters into the opening of the uterine tube by the fimbria. And then the ovarian tube consists of epithelial cells containing cilia on its surface. They continuously beat, and moreover, there is peristalsis or muscular activity going on in oviduct that pulls the ovum inside. In the oviduct, the fertilization occurs. And now this secondary oocyte, do remember this is still stuck in metaphase 2 of meiosis 2, a metaphase 2 of meiosis 2. This remains viable about 1 to 2 days, mostly for 1 day, and if it is not fertilized in this critical window, then it will be shed off and the next cycle will continue. As it is ovulated in the middle of the cycle during the fifth pain, so there may be some pain. So these are called Mittelschmerz pain. These are the mid mild pain during ovulation. So some females have very sensitized for this pain, and they know when ovulation is occurring. So once in the tube, the cumulus cells withdraw their cytoplasmic processes from the zona pellucida and lose contact with the oocyte initially. So as you can see in this picture, here is the primary oocyte, multi-layered stage. Here is the antral stage, here is the graphene stage, and here is the ovulation stage. And here you can see the ovum surrounded by zona pellucida, surrounded by these granulosa cells. So these are called cumulosophorus. So this is the product that is comes out. Initially it gets stuck here, but these are the finger-like projection fimbriae that sweeps across the surface and pulls it inside in the uterine tube. And the follicle that is left behind, that is now called the corpus luteum. Again, these are all pictures from Langman's embryology. This is again from the Langman's embryology, and you can see now you have to figure out, you have to memorize this particular step. So what is present in the ovulated outcome? There is secondary oocyte, there is secondary oocyte stuck in the second meiotic division in the metaphase. So this is secondary oocyte in the metaphase division. Of course, it will have one polar body with it. The is shown right here. In the red, there is zona pellucida, and around it the granulosa cells, now called cumulosophora cell. You will not hear the word cumulosophora. You will not hear the word granulosa. Further on, you will mostly use the word corona radiata, but cumulosophora cells, the one that will be shut off, are still there. So corona radiata, in some books they are interchangeably used. So these cells are there. And then you have this follicle in the yellow. You have theca interna and externa, and then you have granulosa cells, and you have antral cavity. So you had two meiotic arrest points from the fifth intrauterine life month of intrauterine life onwards. All of those primary oocytes, all of the primary oocytes were stuck in prophase 1 diplotin stage of prophase 1 of meiosis 1, and it remained all the way till the growing follicle reached the point of ovulation, and this was further triggered by LH surge. And so the minimum age of this release is at the puberty for those of oocytes that are released at that time, and this arrest is maintained in those primary oocytes that go all the way till menopause. So let's say a woman is having a menopause at 58 years of age or 54 years of age. So that prophase 1 diplotin meiotic arrest remained for that age. And metaphase 2 arrange, and that starts with the formation of secondary oocytes after LH surge, uh, by before ovulation, and this remains there till after fertilization, and only when sperm fuses then it goes beyond that. For all the other females who are having their regular cycles, who are not getting pregnant, so the released ovum in their menses is in the metaphase 2. So the unfertilized ovum is in the metaphase 2 arrest. So these things are shown. This is a picture from both are pictures from Langman's embryology. You have primary oocyte in the division, then you will have this meiosis 1, and you will have secondary oocyte with first polar body, and this will be in the arrested in the metaphase. This will remain arrested in the metaphase. This is the picture we did before in the previous lecture. Primary oocyte, the one that is formed by mitosis, it is diploid. In interphase it becomes diploid, double structured, duplicated, and then it goes through first meiotic division, meiosis 1, and this ends up as secondary oocyte, and it is haploid and double structured, that is duplicated. And if it fuses with sperm, it goes through meiosis 2 or second maturation division, and it leads to mature oocyte, and that is haploid and non duplicated. So you will see it has single chromosomes. So after ovulation, you are at metaphase of meiosis 2. I just made a little slide for slide comparison. I know there is too much information given up till this point, so the kettle thing is little lighter. So just let's, you don't have to memorize any of this, just to give an idea. So this is primordial follicle, about 25 micrometer. It grows so big to primary oocyte, unilamellar stage 18 micrometer, that grows all the way to 125 micrometer in the I have made the relevant ratios are right, so it becomes 125 micrometer, and then you get secondary oocyte. When you have antrum in there, you have follicular loculi, liquor folliculi is present, then secondary follicle, and that grows to all the way till 1 to 2 centimeter. I have 1 centimeter 10 millimeter here for the Graafian follicle. It can be double of that too, that is 20 or 25 millimeters. So that is a general idea. So you can see how big the follicle goes through when it goes from its different stages. So when there is ovulation, the part that is left behind, the theca interna, the theca externa, the granulosa cells, the this collapsed follicle that goes there, this is now organized into corpus luteum. Cells of both granulosa cells and the theca interna, they change their histology and they function under the influence of luteinizing hormone, now the dominant gonadotrophin at this part of the cycle, and now they are relatively yellowish in appearance. They get lipochrome pigment in them. That is why that is called corpus luteum. Luteum is for yellow, and now they develop to be specifically they develop to be steroid producing cell, and their primary function now becomes the progesterone production. This is corpus luteum. The fate of the corpus luteum depends whether the fertilization occurs and where the pregnancy occurs or not, or it does occur. In case of females, so if the pregnancy does not occur, there is no fertilization, and so what happens is that, so after ovulation surge, this LH surge, it converts this LH surge not only helps the ovum to finalize its structure, but it also helps the conversion into corpus luteum, where the cells become steroid producing cells and they become yellow and swollen up and all that. So the LH surge, as long as this LH surge is maintained, this maintains this corpus luteum. So that occurs usually for 10 to 12 days, sometimes 14 days. So after 12 days, this LH surge, LH level decreases. As you can see, the ratio between LH and FSH, it is inversed. And when that support is gone, the corpus luteum starts to go back. It starts to regress, and ultimately it forms a small white scar tissue, and that small white scar tissue is called corpus albicans, and that is ultimately this was producing progesterone. If this is decreased, this is completely regressed, this stop producing progesterone, and then it triggers the menstruation. So what happens if the pregnancy occurs? If the pregnancy occurs, I will study in the immediate lectures that the pregnancy will relate to development of trophoblast, embryoblast, and trophoblast will develop syncytial and cytotrophoblast. So the product of fertilization, the conceptus, has a layer called syncytotrophoblast, and that secretes human chorionic gonadotrophin. When human chorionic gonadotrophin produce, it acts similar to the LH. The HCG, it stimulates the corpus luteum, and this stimulates corpus luteum. It means the corpus luteum sustains. Not only it sustains, it grows. It grows big. It grows so big that it becomes so large that it covers the whole of the ovary. About one third to one half of the ovary is covered by this corpus luteum of pregnancy, and it is maintained by human chorionic gonadotrophin for by HCG. So HCG maintains corpus luteum. Corpus luteum produces progesterone, and progesterone maintains the uterine mucosa and pregnancy. And within these 4 months, if the uterus is removed, if the corpus luteum is removed, if the progesterone supply is interrupted, then the pregnancy drops. After 4 to 5 months, the placenta is developed and matured, and that started to secrete adequate levels of progesterone, and then it degenerates and replaced by corpus albicans that occurred early in the case of non-pregnant case. So that is corpus luteum having the different fates between uh, depending upon the pregnancy occurring or not. So I'm not discussing those in detail in here. This is a picture from Jen Coira's textbook of histology, and here the ovum is compared with the follicle. So before birth, in the shown in the blue here, you have oogonia. Ultimately, near birth, all of them converted into primary oocyte, arrested in prophase meiosis 1, and all of those are surrounded by follicular cells. So all of these are in primordial follicle. After birth, before puberty, there is status quo. Yes, their number decrease, but the oocyte remain in primary oocyte, the follicle remains at primordial follicle. The change now occurs only when after the menarche, when the reproductive cycle starts, and for the selective follicles, it changes from primary to secondary to antral to mature follicle. So follicle goes through all those maturation stages, and the oocyte through all these stages remain in the original stage of prophase 1 of meiosis 1 diplotin stage. It goes only in the meiosis 2 metaphase just before ovulation. So it goes into that. You have polar body and secondary oocyte stuck in metaphase 2 of meiosis 2. So this occurs before and 2-3 hours before ovulation. This is ovulated, and this remains in the ovulatory stage in metaphase 2 until the sperm comes and fuses with it, and then the meiosis 2 is completed, and thus cleavage and the next cycle starts. And after the ovulation, the follicle becomes corpus luteum and corpus albicans. So this is about oogenesis. We talked about this and this. You can pause the video here and go through them in detail for yourself. So this is a picture from Langman's Embryology, second South Asian edition. So I'm not discussing with you this further, but you can pause the video here and take a screenshot regarding about intrauterine life. Third month, you have a large number of primordial germ cells. In the fifth month, especially converted to primary oocyte. Mitosis is going on, increasing their number, and they are converted into meiosis 1. They are starting to get arrested in prophase 1. By seventh month, all of those have been converted to primary oocyte, and all of them have been surrounded by follicular cells to form primordial follicular, and their number is constantly decreasing. At birth, from 7 million, it went to about 700,000, one tenth, 90% dead. Still, they are primary oocyte and primordial follicle, and they are arrested in diplotin stage. Till puberty, still 120 remains, 1 by 20 remains, 95% die, 40,000. And they, after puberty, the oocyte goes to secondary oocyte at the time of ovulation, and meiosis 2 is completed before 3 hours before ovulation, and fertilization after that. The secondary oocyte fertilizes, and it becomes zygote after completed in meiosis 2. Abnormal follicles. Normal follicles consist of more than one cells or multinucleated cells. Oh, there can be a large number of abnormal cells. So I think we are nearing the end. We have discussed in detail the for the ovarian cycle from the perspective of follicles, the growth of the follicles, the growth of the ovum, and the state of FSH and LHN during this process. Now we'll discuss the female reproductive cycle as a whole, and now we'll compare it with the estrogen and progesterone and the endometrial phases too. So female reproductive tracts consist of the uterus with a small tube at the end called cervix, and the tube below that called the vagina, and then these are tubes that connects the uterus to the ovum, that is called uterine tubes or oviducts, that further divided into intramural or uterine part, isthmus part, ampulla part, and infundibular part. Infundibular part is the one that contains this fimbria, And now they have broad ligaments. So the anatomy of broad ligament, the blood supply, the structure, the positioning, the support, all of these things are discussed in the lectures in the gross anatomy of pelvis and perineum. So refer to those lectures for that. So, female reproductive cycle is basically the cyclic changes that occurs in ovary and uterus. It prepares for fertilization, pregnancy, controlled by reproductive hormones. It can be seen from different lenses of follicle, progonadotrophins for hormones, and endometrium. This is, by the way, picture from Jen Coera's histology. So follicular and luteal phases. In the follicular, we had all the follicular phases going all the way from primordial follicle to up till mature follicle, Graafian follicle. Then there was a point of ovulation, and then there was luteal phases. There was corpus luteum, and it regressed to form corpus albicans. Granulosa cells helped in developing oocyte, produced estrogen, endogens to estradiol, and theca cell around them produced androstenedione and testosterone. Then there was gonadotrophin, FSH, and LH. FSH was primarily responsible for triggering and maintaining the follicular growth phase, and they are released in term by due to influence of gonadotrophin releasing hormone from hypothalamus that starts at puberty, and that leads to FSH and LH. And this maintains from this to this. FSH maintain follicular phase. Then there is LH surge, and that produces ovulation. And then there is a relatively higher level of LH continuously decreasing, but it maintains corpus luteum till it lasts. Then estrogen and progesterone. When follicular stimulating hormone is maintaining a pool of follicles, and follicles are converting into granulosa cells are producing, and one theory is given that the large number of follicular cells that are incorporated, they may have an important role in maintaining the estrogen levels in the body, because those granulosa cells, they produce estradiol, and those estrogen are produced from the granulosa cell that goes into the blood and increases the level of progesterone. Extensive detail is mentioned in the physiology reproductive cycle lectures, but basic thing in the initial levels, it is a positive feedback cycle, include helping out the follicular stimulating hormone. But when it raises beyond a particular point, when granulosa cell becomes too much and the estrogen levels are too much, then this estrogen surge, this is the one that is responsible for estrogen surge is responsible for stimulating the luteinizing hormone surge, and that is responsible for ovulation. Then you will get to corpus luteum, and after corpus luteum is the one that is producing the progesterone production, and after the ovulation you have corpus luteum and the high level of progesterone, and when corpus luteum starts to digress, the levels of progesterone starts to drop too. So, because also progesterone production gives a negative feedback on to gonadotrophin releasing hormone, so that results in low level of LH, and ultimately it closes itself. So, high progesterone to low GnRH, low LH to corpus luteum low and progesterone low, and ultimately the progesterone level falls below a critical point where menses start. So endometrial changes can be divided into three basic stages. Before going back to the endometrium, I just wanted to share this with you again. You have mentioned we just I wanted to compare the point of ovulation surge. This point, this is the point of ovulation surge, and this is the point of LH surge, and this is the point of ovulation. So you can see they are successively triggering one another. LH was low, you got you get the follicles grew to the maximum size. They lead to high level of estrogen. Estrogen led to, after a day, it led to LH surge, and LH surge led to ovulation, and ovulation then led to, so each thing is helping out the next one in the pathway. So let's see what was happening in the endometrium. So let us start with the day one of the menstrual cycle, that is the bleeding day one. that occurs when the progesterone falls below a critical point. Then this progesterone is unable to support the endometrium that has been thickened, and so the endometrium sloughs off, the extra glands, the extra blood vessels, the extra thickening of endometrium, all of it breaks off and it comes out. And that is called the bleeding, the menstrual flow, the menses, and it remains for a few days, 3 to 5 days. The menstrual phase is going on where all of the extra endometrium is shed off. So at the end of menstrual phase, the bleeding stops and you are left with a broken up, torn up endometrium, and this endometrium is now repair stimulated by estrogen. As you can see, where this has occurred at the time, you can see where progesterone fell below a critical point. You see here progesterone fell below a critical point. At this point, the bleeding started, and after the bleeding started, so the estrogen levels are high. So estrogen level, they are starting to build up, and estrogen levels are building up. At the one end, estrogen is going to stimulate the follicular development. At the other end, it is going to repair the damage that has been done during menses. So this is the this is the proliferative phase. Now this phase was called follicular phase with respect to the maturation of follicle as it was going on, but here it is called the proliferative phase because endometrial stroma or endometrial epithelial lining of the endometrium, that repairs itself, that divides, that comes back, and the epithelium continuity is maintained and thickness is maintained, and so this is the proliferative phase and it gets back healthy again. So the endometrium now consists of basal layer. This is the basal layer. This is the one that does not shut off. The basal layer does not shut off during menses, and the all of the upper layer, the functional layer, that does shed. The functional layer, this is the basal layer, and functional layer is further divided into compact layer, lightly shown at this point, and the other spongy layer on the top. Compact layer on the top, sorry, compact layer. So this is wrong. So compact layer is near the top. So this is the compact layer on the top, and the one that is between them is the spongy layer. And the cis spongy layer, it is continuously dividing the spiral arteries, forming the spiral arteries and the glands that are present between them. And then the ovulation occurs at day 14, and at day 14 ovulation occurs, and as we saw that after ovulation you have a surge on progesterone hormone, and when this progesterone hormone surges, that is a stimulating factor for endometrium a lot. So you have endometrium greatly increasing in thickness as it was at the end of the secretory phase. So endometrium increases in thick, the spongy layer thickens, you get edema, you get a large amount of fluid, you get dilated, tortuous bodies of uterine glands. So they were such healthy that they are tortuous and they fold upon themselves because they have no place to go. The arteries, they become very tortuous and big and spiral arteries. So all of these things are happening in the endometrium, the secretory phase, and that is making the endometrium ready for implantation. And then again we get to this point where you have progesterone falling below a particular point, then menses start again. So these are the different summary of the different phases. You can pause the video here and take the picture. Proliferative one was where the endometrium was being established, and lengthening of the spiral arteries, fallopian tube had epithelial cell hypertrophy, secretion, cilia is beating, cilia genesis is being done, and mucus plug thins to allow to passage of sperm for this fertile period, the oncoming fertile period, the ovulation. The endometrium thickum, nutrient deposits are there, implementation is here, the oocyte propelled by ciliary activity after ovulation by peristaltic movement towards the oocyte, cervical glands enlarge. This is triggered by majorly LH cells and then edema of endometrium, glycogen deposition, tortuous uterine gland, increased vascularity through spiral arteries. And this is occurring in the endometrium, and the fallopian tube is rolling back. Ciliation is deciliation, secretion is being reduced, and the mucus plug is being thickened again to prevent more sperm entry that is occurring through the progesterone. I have not mentioned in this particular table the menstrual phase, and that is due to the decrease in the level of progesterone, where everything falls apart, and apart from basal layer, everything shuts off. So this is the overall picture. I think I've summarized it with it again. But if any question still remains, please mention in the comments below. I will try my level best to answer. So you have, and one more thing, from exam point of view, uh, the examiners may be little frank with the use of phases interchangeably. So in an MCQ regarding the mention of where implantation occur, rather than getting a secretory phase and an option of the MCQ, you may get a luteal phase too. So there may be some questions here where the things are mixed. So do pause the video here and go through these comparisons. If you can do that, you are golden. This is a picture from Langman's embryology, denoting the same things. Thank you so much. I know it has been a heavy lecture. I'm trying to make it as simple as I can, but that extends the amount of lecture. And I'm trying to break up these lectures into chapters, and you can directly jump to those. If you have further questions, suggestions, and feedback, please mention in the comments below. You can access us through our website, through our Facebook page, or YouTube page, through our Instagram page. For any updates, you can contact us personally too. And do follow us for this complete series of embryology, and explore our pages for the other subjects going on for physiology and biochemistry for the whole course we are taking from the start. So, good luck. Thank you so much. See you in the next lecture. Till then, Allah Hafiz.